Good evening and welcome to this Retina UK information webinar. This is one of a series of webinars that we're hosting and we'll be delivering at least one on a different topic each month. Today we're really pleased to be joined by Roly Megaw. Roly is a Wellcome Trust funded clinical lecturer at the MRC Human Genetics Unit at University of Edinburgh and a consultant ophthalmologist in NHS Lothian. Roly undertook his PhD with Charles French Constant at the Scottish Centre for Regenerative Medicine. Using induced pluripotent stem cells to model X-linked retinitis pigmentosa prior to taking his clinical fellowship with Graham Black in Manchester. Roly now runs the Inherited Retinal Dystrophy Service in Edinburgh, allowing him to combine his clinical and research interests. We're also joined today by Matt Carr, our Information and Support Manager, and Kate Arkle, our Research Development Manager, who'll be collecting questions throughout the presentation. Now, there are a couple of ways for you to ask questions. You can either type your question in the Q&A section at the bottom of your screens, and these questions will be asked by the team on your behalf. Alternatively, you can raise your hand by pressing the Alt and Y keys if you are using Windows computer, or the Option and Y if you're using a Mac. We'll then be able to ask you to unmute your microphone in order for you to ask your own question. So please do leave your questions throughout the presentation and they'll be answered at the end. We'll endeavour to answer all of your questions, or certainly as many as we can, but any questions we're not able to get around to today will be followed up over the course of the next couple of weeks. Thank you again for joining us, and without further ado, I'm delighted to hand over to Roly.
Thanks very much, Denise. Denise, can you just nod if you can hear me now that I've unmuted? Yeah, excellent. Okay. All right, good evening, everybody. I'm going to share my screen, which you can hopefully all see now. Again, Denise, you just want to give a quick nod if, yeah, excellent, thumbs up, fab. Okay, great. So every day's a school day. I had no idea that Command and Y raise your hand in Zoom, but now I do know. That's great. Thanks so much for inviting me, and thanks so much for coming along to listen. I'm sure I've presented to a few of you in the past. I've done a few talks for Retina UK. I've done a few talks for RP Fighting Blindness back when it was RP Fighting Blindness, and I also present quite a lot to the Edinburgh group that well, pre-COVID used to meet every few months up in Edinburgh. So I, for those who I haven't presented to or met before, as Denise says, my clinical and research interest is in retinal dystrophies, and mainly RP. And the vast majority of my research has always been trying to understand the mechanism that which genes which cause RP, or rather genes mutations in which cause RP, I've always been trying to figure out the mechanism as to how they function within the cells at the back of the eye. But latterly, I've begun to think a little bit about how cells die in retinitis pigmentosa, and we've been doing a little bit of work in the lab on that, and I thought rather than just churn out the same old mechanistic story that I normally talk about, maybe I could talk about the new work we've been doing, which is trying to understand how photoreceptors die. And the reason for that, and the reason I always say about understanding mechanism as well, is that we are really on the, hopefully, the precipice, the start of a gene therapy revolution. We, as you all know, we now have a gene therapy in clinic for patients with mutations in the RPE65 gene, who then develop either Leber's congenital amaurosis, which is a retinal dystrophy, or they can develop quite severe retinitis pigmentosa. And so that has finally made it to clinic. We can give it in the NHS, and there now follow several gene therapy trials which are getting up and running to target different genes in RP, and more will follow. And so we are really at the forefront are at the start, hopefully, gene therapy becoming a real treatment option for patients with RP. But that said, the improvement in vision that the trials have shown, the published literature shows that whilst there is definitely an improvement in vision whenever you give gene therapy, the vision doesn't improve to to a 20/20 level, and therefore there's always room to give something else. Whenever someone has high blood pressure, they tend to start on one drug, but inevitably they end up on 2 or 3 drugs. And I very much think that retinitis pigmentosa and retinal dystrophies in general will be eventually, hopefully, if all our research pays off, will be treated by a sort of a polypharmacy, just like high blood pressure, as an example is. So yeah, so I'm going to talk to you tonight about what we've been doing to try to better understand how cells die in retinitis pigmentosa, because if you can stop cells dying, then in theory that will allow successfully what we call transfected cells, cells that have been given a gene therapy. Hopefully that will allow them to function for longer if you're also stopping them dying. So just before I start, I sometimes like to start with this slide in my talks. What I'm showing you here is the euglena, which is a single-celled flagellate organism. So it swims around in water, and it was around during the Cambrian explosion about 541 million years ago. And it was a single cell, and it floated around, and the only way it could interact with its surroundings was by touch and feel. And then one day, one of the euglena had a mutation in one of its genes, and it started producing a protein because genes. And because of that, you could then suddenly, or these euglena could suddenly appreciate their surroundings. They could, light would fall on these cells, on these little organisms, and the fluorophore, this light-sensing protein, would change shape, and therefore they could sense light. But they had nothing really, they couldn't really process this information. So they then had to sort of build a central processing unit behind this primitive eye, and that became the brain. So as we like to say in ophthalmologists, and no matter what neurologists say, we like to say that the brain is simply an extension of the eye. I don't think anybody here needs convincing that the eye is the most important organ in the body, but I just like to get that in there early, that the light sensing part of our body, the eye, was around way before the brain. And here we have the eye, the modern day human eye. I'm showing you just a cross section of a cartoon of an eye. Light shines through the cornea, which is the crystal clear window at the front of the eye. It then moves through your pupil and is what we call refracted by your lens. So your lens changes shape to allow you to focus on light that's coming from near objects and distant objects. And it's that lens that becomes cloudy whenever you get a cataract. And obviously RP very much causes early onset cataract, and we remove that lens and replace it with a little plastic lens in cataract surgery. And then the light falls on the business end of the eye at the back of the eye, which is the retina. And the retina is a 3-layered neurosensory structure, which is just absolutely beautiful in its simplicity. It's a 3-order structure. So light sensing photoreceptors communicate with bipolar cells, which communicate with ganglion cells, and that's all it takes to transmit an a signal from light into an electrical signal, which allows you to see. So it's an absolutely incredible structure, and so very simple. And as I mentioned, right at the back, we have the light sensing photoreceptor, which in my opinion is the most important cell in the body. And you have about 120 million of these in each eye. Now, the photoreceptor has evolved to contain, it's made up of an inner segment and an outer segment. Now, this outer segment is really just a modified cilium. Now, the cilium are the antennae of cells which detect signals. That they're the signaling organelle of every cell, and cilia detect signals, or they signal because of a protein called a G protein coupled receptor. And our light sensing pigment that the euglena first developed 540 million years ago is a G protein coupled receptor. And in order for normal vision to occur, these G protein coupled receptors called rhodopsin have to be packed into the outer segment, which is an expansion of the cilia membrane. And so there's a constant turnover of rhodopsin. Thousands of molecules are trafficked from the inner segment of the photoreceptor into the outer segment, which allows them to be packed together. And therefore, whenever light falls on the retina, it stimulates these rhodopsin molecules in the outer segment, which then allows us to generate an electrochemical signal, which then goes to the brain and tells us what we're seeing. So this photoreceptor is the most important cell for vision. It is very, very active because of the thousands of molecules per second that have to be trafficked from the inner segment to the outer segment. And therefore, they're very susceptible to things going wrong. And of course, whenever things go wrong in the photoreceptor, patients get retinitis pigmentosa. And retinitis pigmentosa is the leading cause of visual loss in children and in adults of working age. And what I'm showing you in this slide is a healthy retina on the left and a retinitis pigmentosa retina on the back. So whenever we look at the back of people's eyes in clinic, this is classically what we see. We see peripheral bone spicules, pigmented bone spicules in the periphery of the retina. Hence the name retinitis pigmentosa. We see thinning of the blood vessels, and we see the optic nerve going pale. And whenever we look at that, we see it's retinitis pigmentosa. And patients with this, whenever they start developing this, as all the patients today, all the listeners today with RP will testify, they get loss of their night vision, constriction of their visual fields, and eventual loss of their central vision. But this is, retinitis pigmentosa is a fairly archaic term. It's fairly old fashioned. It's, we diagnose it whenever you look at the back of the eye. But in actual fact, mutations in over 150 genes cause the same clinical picture. And so the last 30 years have been spent by geneticists and researchers trying to identify all the different genes which contribute and cause retinitis pigmentosa. And now that we know of the vast majority of these genes, our job now is to better understand their function and better understand how these cells are dying. So 3 approaches to identifying treatments that we work on in the lab is, number 1, as I mentioned, trying to understand what's going wrong and trying to fix it. A second way is to try to stop the cells dying. And a third way is try to fix or replace the gene. And so these 3 approaches, the first one, understanding what's going wrong and fix it, you need to understand the function of genes. And I've spoken with Retina UK about this in the past, about how we do this in the lab. Secondly, as I'll speak about tonight, stopping the cells dying. And in order to do that, we have to understand how the cells are dying. And finally, fixing and replacing the gene. And that is, conventional gene therapy or CRISPR therapy, which I'm not going to speak about today. So as I say, over 150 genes are known to cause it. I'm just showing a graphic here of all the different genes that are commonly tested on panels of genetic analysis whenever patients present with RP and we send their blood off, for example, to the labs in Manchester. These are all the genes that are tested. And my gene of interest, or certainly during my PhD and into my postdoc jobs, is the retinitis pigmentosa GTPase regulator gene, or RPGR, which causes the vast majority of X-linked disease, about 90%. It causes about 15% of all RP, and it's a very aggressive form of disease. Patients get visual loss early on, and so it causes quite a severe form of retinitis pigmentosa. So, like I say, laterally we've been thinking, how do photoreceptors die in RP? Do they all die the same way? Just in the last slide, I showed you this list of 150 genes. And for gene therapy, we need to target those genes one at a time. But actually, if photoreceptors all died in the same way, if the final downstream common pathway of cell death was the same, then perhaps if you could understand it and identify a drug which could stop it happening, then this one drug could treat multiple alleles, multiple different genetic causes of RP. And what I'm showing you in this slide is just a little cartoon which Faye Newton in the lab produced in a review article about photoreceptor death in RP. And it just shows the complicated nature of photoreceptor death, how poorly understood it is, and how several different pathways have been proposed for how photoreceptors die in RP. Traditionally, it was always thought that cells died by a cell death mechanism called apoptosis, which is a programmed cell death. And by programmed, I mean the cell itself decides to die because it's not functioning properly and releases a series of chemicals within the cell which cause it to apoptose, to shrink, and to sort of melt away. But latterly, other forms of disease cell death have been identified as possible causes of photoreceptor death in RP. One of which is necroptosis, which is again another form of programmed cell death, but it is much more akin to necrosis, which is unprogrammed cell death, where the cell effectively bursts and releases all its contents into the tissue in which it resides. And if necroptosis is the cause of cell death in RP, it would release a lot of pro-inflammatory molecules, which would lead to inflammation in RP. And we know that retinitis pigmentosa has a very low-grade chronic inflammatory component because the type of cataracts you get in RP tend to be what we call patients with inflammatory eye disease get. We know that sometimes in the back of the eye you can see very low-grade inflammation in the eye. And we know from studies that if you remove samples from the eye of patients with RP and do very simple analysis of what we call cytokines, which are molecules which regulate inflammation, there are pro-inflammatory cytokines in the eye of a patient with RP. So this would be in keeping with necroptosis being a cause of cell death in RP. So do photoreceptors all die in the same way in RP? And if they do, can the cell death pathway be a target for therapy? And that's what we've latterly been thinking. So as Denise said, I did my PhD using stem cells, but sort of the work I've been doing in the last 5 years or so, I've actually been working on animal models because in many ways they're better models to assess photoreceptor function. And so we generated some new models of RPGR mediated RP. So we took some mice and we mutated their RPGR gene. And in the lab, in the human genetics unit where I work, we've got a very nice phenotyping suite where we've, over the years, managed to accrue much of the equipment that we use in clinic to diagnose RP and to monitor the progression of RP. And we have that now in the HGU to look at our mice. And one of the things we have is an electrophysiology machine. Electrodiagnostic testing is something which is the sort of the absolute ultimate way of clinically diagnosing RP. A lot of you will no doubt have had this done. You have electrodes attached to your eyes. You are sat in the dark for 30 to 40 minutes, and then very uncomfortably lots of flashes of light are flashed at you. And that allows us to assess the electrical function of the retina, and we see very characteristic patterns of RP, characteristic patterns of changes in your electrical function with RP. And we can see that in our mice. On the left, what I'm showing you is a photograph of our mice in the machine. And below that, I'm showing you tracings of the electrophysiology readout. In blue, you get the classic trace. And in red, I'm showing you the tracings of the mutant mice, which have a flattened ERG curve, which shows that they're not seeing as well. We also have an OCT machine, so anyone who's been to the opticians or indeed to an eye hospital will no doubt have had their backs of their eyes imaged by an OCT machine, which takes a lovely, almost like a histological cross section of the back of the eye and allows us to look at retinal thickness. And what I'm showing you in the middle here is a photograph of a healthy mouse, and below that, one of the mice with mutations in RPGR. And what I'm showing you is that there is thinning of the retina as these photoreceptors die. Another really good marker that we use in clinic for assessing progression of disease and even diagnosing disease is autofluorescence, where we shine a blue light into the back of the eye. And whenever the retina is becoming a little bit unhealthy, the retina autofluoresces normally. But whenever it starts to become unhealthy, it autofluoresces, it has an increase in autofluorescence. And what we see in our mice, and what I'm showing you on the right, is that the RP model, the RPGR mutant mouse, gets increased autofluorescence. So these are lovely tools of assessing loss of function and loss of structure and ill health in RP, and therefore we can monitor mice and see whether they are losing sight and whether we can prevent it with drugs. So in order to better understand how cells are dying, we have been using a technique called single cell RNA sequencing. Now, sequencing is where we look at either the DNA or the RNA to see if we're looking at the DNA, we sequence the DNA, which is, and when I say sequence, what I mean is we read along the genetic code for all the different letters that make up the genetic code. And whenever we sequence people, the genes of people with RP, we can find changes in the sequence, and that and these changes are disease causing. Now, DNA makes RNA and RNA makes protein, and we can also sequence the RNA in samples. And whenever we do that, we can see it's a good marker for the amount of RNA that's being made. We can see levels of RNA, and changes in RNA levels between healthy and unhealthy tissue can give you ideas as to, well, many things, including just how unhealthy a cell is. And what we have always done in the past is we've done bulk RNA sequencing, where we've just taken a load of cells or a load of tissue and we've extracted the RNA from all the tissue, and we've run that on a sequencer, and we've detected how much RNA is being made in the entire tissue. But unfortunately, the retina is made up of 6 different cell types. That's actually quite lucky that, most tissues, for example, the brain, are hundreds and hundreds of cell types, but still 6 is too many, because you've got the light sensing photoreceptors, but you've also got bipolar cells, ganglion cells, amacrine cells, retinal pigment epithelium. And so the RNA that's being produced by all those cells muddies the water and doesn't allow you to actually understand exactly how much RNA is being produced by the photoreceptors. So the new technique is called single cell RNA sequencing, which allows you to analyze the RNA that's being made of every single cell individually. And it's an incredible technique, and I'm just going to talk you through it slowly. So what we do is we take a retina of our RPGR mutant mice and of our healthy mice, and we dissociate the retina into all the different cell types. What we then do is we put it on a single cell chip. And this, by doing so, every single cell is put into a little microgel micro droplet of gel, and the cells are then lysed, and the RNA is harvested from these cells. And so every single cell is is separated, and therefore the RNA from each individual cell is put in this microdroplet. What then happens is all the RNAs, all the different transcripts, so if you remember, DNA makes RNA, makes proteins, so the cell uses DNA to make all the RNA, and there are different levels of RNA, and each RNA transcript is then tagged with a barcode, which is a sequence of DNA. It's a string of letters that's unique to that microdroplet. So every single RNA transcript has a barcode, and therefore it can be traced back to that individual droplet and therefore that individual cell. Then we amplify the RNA, so we make tons of or rather we turn it almost back into DNA and then amplify it. So then you have millions and millions of transcripts, but again, they're all barcoded, so we know exactly which cell they came from. And you then read every single transcript on a sequencer, and you get hundreds and hundreds of millions of reads, of transcript reads. And from those transcripts, from those sequence, you know what gene has made that RNA, and therefore what protein will that RNA will go on to make, and therefore you can get a good understanding of the levels of the proteins being made in every single cell, because like I say, you can then map it back to each individual cell. And what we get, and what I'm showing you in this slide, is we get a plot of every single cell that we have, analyze the RNA, and we use we use software to cluster these cells. So just if we just talk about photoreceptors, one of the most important proteins and one of the most common proteins that each photoreceptor makes is rhodopsin, because it's the light sensing cell. So if you have 10,000 cells and 8,000 of them are making lots of rhodopsin, you know that those cells are photoreceptors, and you can, because we understand the different genes that different cell types make, we can then separate all these cells, and we can label these cells. We know which ones are photoreceptors, which ones are bipolar cells, and which ones are ganglion cells, etc., etc. So what I'm showing you here is a t-SNE plot of all the different cells in one of our experiments. And the cell got 20 different clusters. And by looking at the RNA levels, we are able to tell which ones are the photoreceptors. And what I'm showing you here then is just a blown up version of every single photoreceptor cell that was harvested in the experiment and was analyzed. And we can then look at the not just the rhodopsin RNA, but we can look at the levels of every single gene that it's been making. And we can we can analyze the photoreceptors, and we can see a disease trajectory, because the photoreceptors make up 4 different clusters. So there are 4 groups of photoreceptors which are making different amounts of RNA. And on the left, what I'm showing you is the red cluster, the yellow cluster, the green cluster, and the blue cluster. And the red cluster, there were, because we've barcoded these, we know exactly which cells are and which mice these cells come from. And the vast majority of the photoreceptors in the red cluster are from the wild type healthy mouse. And the vast majority of the green and blue clusters are from the RPGR mutant mouse. So they seem to separate out. And whenever we look at the RNA profiles of these different clusters, what we see is in cluster 1, we look at the differences between the RPGR mutant and the wild type mouse, and we see that as the cells move from cluster 1 to 2 to 3 to 4, they start making less genes or less RNAs that are involved in phototransduction. And what I mean by that is light detection. So we think these cells are getting sick, and they're stopping making the proteins that are needed for light sensing. And as I said, these ones in clusters 3 and 4, the vast majority of these cells are from the RPGR mutant mouse. So that's very interesting. So it shows that as they start moving from health to stress to disease to death, they start down-regulating, they start making less proteins that are involved in phototransduction. But what we then look at is we look at the RNA profiles that are, or we look at the different levels of RNA that are upregulated in the mutant photoreceptor. And what we see is that there are lots of different pathways that are being upregulated, but one in particular is necroptosis. And as I said, necroptosis is this cell death pathway that is potentially been shown, there are signs that it might be involved in cell death in RP, and it appears to be upregulated in our dying RPGR mutant cells. So what we then did was, or what we've done so far, is just to go further and start to look at necroptosis and to try to understand whether this is happening in our photoreceptors. So necroptosis is regulated by a protein called AKT. And when when AKT is activated by phosphorylating it, and what I'm showing you here is an analysis of the protein of our RPGR mutant retinas and the wild type, and there appears to be increased phosphorylation of AKT, increased activation of AKT. So that would be in keeping with what leads to necroptosis. And whenever we look at the cells themselves, whenever we make very, very fine sections of the back of the eye and look at a very high power scope, what we see in our receptors, light here showing you is an electron microscope of a burst dead photoreceptor cell, and rather than apoptosis, rather than this programmed cell death, what we're seeing is a burst cell, which would be more in keeping with necroptosis. What we then did was look at a protein that is required for necroptosis. It's a key marker of necroptosis. It's called MLKL. And what I'm showing you on the left here is 6 photographs, 3 of the wild type eye, 3 of the mutant eye, and I'm showing you 1 each at 6 months, 12 months, and 18 months. And in labeled in fluorescent green are the MLKL positive cells. And in the mutant cell, as the mice gets older and as the cells start to die, more and more of these cells are positive for MLKL, which is this protein that is required for necroptosis. And what I'm showing you on the right here is a graph showing that the number of cells that are MLKL positive goes up and up and up as the mouse gets older and as the photoreceptors start to die. So this would be in keeping with necroptosis as a mechanism of cell death in RP. Now, this is exciting because necroptosis inhibitors are already in clinical trial for various inflammatory diseases and various neurodegenerative diseases. And what I'm showing you here is just a table from a review by Martins et al. From last year. RIP kinases are the which activate MLKL, which I spoke about just a couple of minutes ago. And there are several RIP kinase inhibitors which are under investigation for inflammatory conditions such as psoriasis, ulcerative colitis, and rheumatoid arthritis to see if they can dampen down disease. And they're also being investigated in clinical trials to determine whether these necroptosis inhibitors are effective in neurodegenerative diseases like Alzheimer's, amyotrophic lateral sclerosis, and multiple sclerosis. So the great thing about this is these drugs are already in humans, and they're all phase 2 and phase 1 safety trials. But if they show that they are safe, then we know we can use them in the eye. We know that they'll be safe to use for retinitis pigmentosa. Now, the great thing about delivering treatments to the eye is that it's a fairly isolated organ, but these are systemic drugs that are being given to patients with Alzheimer's and ulcerative colitis and other diseases. So if they're safe in these trials, then we would suspect that they would be safe to give to the eye. The other thing of interest is whether Another thing we're interested in is cells that are recruited to the retina whenever the retina starts to degenerate. So what we saw in one of our clusters is what I'm showing you here is this Tisney plot of the single cell seq data, and a cluster of the cells that we identified only in our mutant mice and not in our wild type mice had high RNA levels that suggested that they were macrophages. Now, macrophages are a cell that that eat unhealthy cells. They're recruited to inflamed, stressed on tissue, and they eat cells which are unhealthy. And so what I'm showing you here on the right is sections of a wild type mouse on the left and an RPGR mutant mouse on the right. And in fluorescent green is a marker for macrophages. And sure enough, whenever we stain our retinas, we see macrophages are being recruited to the outer to the retina in our RP models. And this would suggest that these cells, the photoreceptors, are probably releasing recruiting factors, signals which are asking the macrophages to come into the retina to start, breaking up and eating the stressed photoreceptors. And so this is another focus of interest, because if we can identify how these or what signals these photoreceptors are releasing to recruit the macrophages, it again could be potentially a target to stop cells dying. Now, what we really need to do if we are to fully understand the the cell recruitment pathways, the cell signaling, the recruitment signaling, and to absolutely fully understand the mechanism of cell death in the photoreceptor, what we would love to do is do RNA sequencing of just our photoreceptors. So the great thing about single cell sequencing is we can separate out all the different cell types, and therefore we can just look at the RNA transcript levels of our photoreceptors. But one of the drawbacks is that the depth at which the RNA transcripts can be read is pretty shallow compared to old-fashioned bulk RNA sequencing. And so what we would love to do is to isolate the photoreceptors themselves and do bulk RNA sequencing on the photoreceptors, and therefore get a much deeper read and therefore a much better understanding of the fluctuations in RNA and therefore the fluctuations in protein in our photoreceptors as they start to die. And in order to do this, a very nice little mouse model in the lab where we've tagged rhodopsin with a little protein called SNAP. And SNAP can be tagged by fluorescent proteins. So what we can do is we can inject the eyes of these mice with a SNAP tag, with a fluorescent SNAP tag. It then binds to SNAP, and because the mouse we have, SNAP has been genetically tagged to rhodopsin. Whenever the fluorescent tag binds to SNAP, it's binding to rhodopsin, and therefore it's only binding to photoreceptors. And what I'm showing you here is a little movie of a retinal slice of our rhodopsin SNAP mouse. And in fluorescent orange, moving around over about an hour and a half, you can see what I'm showing is outer segments moving back and forth in our retinal slices. So what I can then theoretically do is inject the mice eyes. Well, first of all, what I'll do is I've crossed the rhodopsin SNAP mouse with the RPGR mutant mouse, and I will then I inject the eyes with the fluorescent tag, and then I carry out the conventional protocol to split the cells up. But instead of going to single cell sequencing them, what I do is I pass them through a machine which can sort by fluorescence, so it can detect the fluorescent cells, which are the photoreceptors which have been labeled with this fluorescent tag, and then I can just sort out just the photoreceptors, and therefore I can get a population of photoreceptors of both RPGR mutant and wild type, and we can then submit those for bulk RNA sequencing to get a much deeper read, to get a much better understanding of the changes in RNA that are occurring whenever these mice have mutations that cause RP. So I'm going to stop there. What we've spoken about today is just a little bit of the work we've been doing in trying to understand how cells are dying, because I do think that if we can understand this, and if we can demonstrate that the cells are dying in the same manner across many different mouse models of RP, then in theory we could possibly deduce from that that the photoreceptors in patients with the varying different mutations are dying by the same way, and we can maybe target them with inhibitors of cell death, just like the necroptosis pathway that we've identified. I've got lots of people that I need to thank. The most important person is Faye Newton, who is the really talented postdoc in our lab, who has carried out all this work to try to understand cell death. I work at the MRC Human Genetics Unit in Pleasantine Mills Lab. As Denise said, I've done a lot of work with the French Constant Lab at our Stem Cell Centre in Edinburgh, and I've got various collaborators across the UK and in the States who have been instrumental in all the work I do. And of course, last but very much not least, all the funders who are foolish enough to give me the money to do this work, and then of course the patients in NHS Lothian who are instrumental in helping me carry out the research that I do. So I'm going to stop sharing now, and I'll be more than happy to take any questions.
Roly, thank you so much. Denise, can you just give me a thumbs up if you can hear me now? Yeah, super. Okay. Roly, thank you for an absolutely fascinating talk. That was amazing on so many levels, not least the techniques that you're using in the lab, which are just absolutely incredible. But also it was really nice to hear, I think, about something other than something very gene specific. We tend to focus a lot, I think, on the very gene specific stuff and what are the genes doing and have we found more genes. And I think it's really refreshing actually to hear a bit about the other things that are going on and perhaps some common pathways that will be applicable to a large proportion of our community. So that was really fantastic. I can't see any hands up at the moment, but we have got a question in the Q&A box. Raj says, please could you explain if there is any connection between RP and myasthenia gravis? Now, as far as I remember, myasthenia gravis is an autoimmune disorder where there's a breakdown of the communication between nerve cells and muscle cells. So yeah, can you comment on that at all, Roly?
So as far as I'm aware, there's no link between myasthenia and RP, but there are some diseases where patients get diseases. So myasthenia gravis, yeah, you're absolutely right. It's a breakdown between the nerve and the muscle. You get autoantibodies that attack the synapses, or rather they attack one of the receptors between the nerve and the muscle, and therefore they reduce the ability of the muscle to contract, and therefore that causes weakness. And how that often manifests is weakness in your eye muscles, and therefore you get patients tend to present with either a drooping eyelid or double vision. And so there's no link that I'm aware of between myasthenia gravis and RP, but there is a link between certain diseases that can cause similar problems to eye movement that can also cause RP, and that they tend to be because of mutations in mitochondrial genes. So right. The mitochondria are the sort of the powerhouses, the engines of our cells. They're actually billions of years ago were living on their own, and then they thought it would be a very good idea to jump on board a mammalian cell and live in our cell because it's easier. And the trade back, the sort of symbiosis is they provide us with energy. So they're actually very, very prehistoric living things that that now reside in our cells, and so they have their own DNA. And the DNA of the mitochondria is inherited from your mum, because sperm, the part of the sperm that gets into an egg, doesn't have any mitochondria in it. So the mitochondria we have is all from our mums. And so mutation, and so they have their own mutations, and those mutations can cause eye disease, one of which can be problems with eye movements, and also can cause RP.
Okay, so would that be picked up if somebody had a genetic test? Would that help to clarify if that was the case?
Yeah, so we know of some genes which cause this sort of association. So yeah, absolutely, you'd be specifically looking for them. So if whenever we send our panel testing to whichever lab does it varies over the regions. There's either London, Manchester, or Oxford now. Genomics England is organised into these three centres for testing for retinal disease. Where am I going with this? Yeah, so whenever we send our blood down to test RP, it's tested on a panel which doesn't test mitochondrial genes. So you'd have to be specifically looking for that.
Right, so presumably that would depend on your ophthalmologist noticing a picture of other symptoms as well, which would indicate there might be a mitochondrial problem. Okay, thank you. We've got a nice comment from Valerie, actually, not a question, but Roly, thank you very much for such a clear talk. It's good to know that you've been busying away while a lot of other projects have been on hold during COVID. So an appreciative delegate there. Does anybody else, I've got a question that I could ask, but is anybody else wanting to raise a hand or type anything into the box? Jason, thank you. Jason says, my son was diagnosed 21 years ago with X-linked RP. How long do you feel it will be before we have a cure? That question we all dread.
Yeah. So, and cure is obviously the loaded word there, because to cure it, you really need to correct the mutation. And I'm sure everybody's heard of CRISPRs. They've won the Nobel Prize, or rather the two female scientists who sort of showed that CRISPRs can edit the genome in mammalian and therefore human cells. They've been awarded the Nobel Prize for chemistry last year. So with CRISPRs, you can edit the genome, you can correct genes, you can edit the genome however you want with CRISPRs, and we use them loads in the lab. They're amazing. We're now starting to explore their use in treating genetic disease, and there is a CRISPR trial for one mutation which causes Leber's congenital amaurosis. So it's the really severe retinal dystrophy, and it's a mutation in a gene called SEP. So watch this space, fingers crossed. It's going to be a monumental effort to get that optimized and working, but you never know, and that would lead to a cure because you're correcting mutation. But 21 years of X-linked RP, when is there going to be a treatment? There is a gene therapy. So there are a few forms of X-linked RP, but as I said, the most common one by far is RPGR. 90% of X-linked RP is caused by RPGR mutations, and so the chances are, sorry, I can't remember the name of the guy who wrote the question.
Jason, it was Jason.
So Jason, the chances are your son's probably got an RPGR mutation, and there is a gene therapy trial starting worldwide for RPGR gene therapy imminently. We're a trial centre in Edinburgh. There are two in Scotland. There are at least two in the UK, in England, sorry. And the trial is aiming to treat 50 patients worldwide. giving an RPGR gene therapy. So, you know, again, watch this space. That's, you know, so if Jason wants to be recruited onto the trial, he would have to get, you know, contact his ophthalmologist. I don't know where Jason lives. You can maybe type in to the chat. But yeah, be seen by your ophthalmologist and be referred to a trial site. I'd more than happily see your son to assess him, but Edinburgh might be a long way away, and there may well be a trial site much closer to home.
I think it's also possibly just worth mentioning as well, Roly, that clinical trials will always have quite stringent criteria. Yeah. So there's always a screening process to go through. I know the trial site consultants will all be very happy to see people, but you just need to be aware that there will be a screening to see if people fit the criteria.
Yeah, absolutely. And what we're finding, because RPGR is such a severe disease, is that a lot of the patients I've screened, sadly, they don't meet the minimum criteria of vision, just because it's such a severe disease. So yeah, no, you're quite right. I should have caveated that. You know, someone will happily see them, hopefully to screen them and see whether they would qualify. And you know, some patients who do qualify don't fancy, you know, it is a trial, it's unproven, we know it's pretty safe, but it is a trial. And so but if you would like to find out, yeah, do get in touch with your ophthalmologist.
Okay. Question from Tracy. Does having high blood pressure or things like allergies that cause excessive inflammation cause the cells to die, so I guess contribute to that cell death process?
So I think the allergy part is allergies tend to be external. They tend to be, you know, mucosa, so they tend to be nose, you know, rhinitis, you know, hay fever, allergic conjunctivitis, and that's all external, and that does not contribute to the inflammation at the back of the eye. So that's that. As far as blood pressure goes, we do know that high blood pressure, so the retina is unbelievably metabolic. As I said, it's really active, and it's got this really rich blood supply behind it called the choroid, which, you know, it gives such a huge amount of blood is supplied to the retina given the size of the organ. And high blood pressure, you know, untreated sustained high blood pressure compromises that structure, the choroid, and it damages it, and it therefore brings less blood and less oxygen to the photoreceptors. Now, I don't know of any trial which shows that RP patients with high blood pressure lose sight quicker, but there's no doubt that your photoreceptors are very susceptible, and so, you know, whenever I see my patients in clinic, the first thing I do is ask about smoking, you know, and I don't chastise them, but because I know how difficult it is to quit, you know, something, you know, so addictive, but you know, smoking is just, when you've got these susceptible cells, you have to stop smoking, you have to eat green vegetables, you have to exercise to keep your blood pressure down. All that is, you're standing yourself, and you're giving yourself your best opportunity to slow the disease process. Like I say, there's probably nothing in the literature about blood pressure and accelerating RP, but it would make sense that, you know, you would keep tabs in it and get it under control.
Yeah. Okay, thank you. Mark says, thank you very much for your talk. Can you tell me what part the RP1 gene plays within the retina?
Oh. What does RP1 do?
Is that something to do? Is it rhodopsin?
So genes were all labeled one to whatever, or, you know, I think up to 50 something. If someone can Google RP1 and what the actual gene is, I might be able to.
I've got a feeling it's RHO. But I can check that. Or perhaps Denise, could you have a look for us? Have you got a phone to hand? Jason, by the way, going back to Jason, he says his son lives in Kent and goes to Moorfields. Did you say Moorfields was a centre for the gene therapy trial?
Yeah. UCL and your GTX. So yeah, absolutely. So if your son goes to Moorfields, yeah, they should phone up and just say you've heard about the trial. And if he wants to be screened, then yeah, they'll certainly do that. So RP1 is a microtubule associated proteins. So so the microtubules are, I spoke about cilia. So cilia are the censoring organelles, and they're they're antennae essentially, and the photoreceptor has this huge expanse of ciliary membrane, and the rhodopsin's all stuffed in there, and the microtubules give it structure. They over, but also along the microtubules you have these little motors called dyneins, which bring proteins all the way up the cilia to the tip, which is where the outer segment is. And so these microtubules are almost like the train tracks on which these motor proteins, sorry, it's kinesins bring it to the tip and dyneins bring them back, but it's along these microtubules. So RP1, I just googled it. It's haven't properly read it, but it looks like it's an axonemal microtubule associated protein. So it goes towards building these train tracks, which give the scaffold for the cell, for the cilia, plus allow the proteins to work their way up to the tip.
Brilliant, thank you. Don, we've got time. I think this might have to be the last one, but Don just says, I've had RP for over 30 years. I recently had a genetic test from a blood sample which found one faulty gene and one possibly faulty gene. Are there any more tests which could be worth having?
So I assume these, so you've got 2 copies of every gene, and the vast majority of RP is, or the vast majority of genetic disease is recessive disease. And with recessive disease, you need 2 faulty copies, because with only 1 faulty copy, the healthy copy can make enough protein that you don't have a disease. And so I'm assuming you find what the test is showing is that you have a mutation in 1 copy of a specific gene, which we know causes RP, and then you've got a change in the other gene, which has never been shown to cause RP before. And so it's called a VUS, it's called a variant of unknown significance, because we don't know if that mutation is damaging to the gene, i.e. does the mutation mean you don't make the right protein, a healthy protein? And so the way they're figured, so if you have a VUS, a variant of unknown significance, it's because it's never been reported in the literature before, and therefore we you know, if 5,000 people around the world had that and they all had RP, we'd say, well, it's clearly disease causing. People can take these, so computational biologists are people that work on a computer, take proteins, mutate little things, and they see and then they predict how the protein shape will be and whether it'll interfere with the function of the protein. And you're going to have to wait, I guess, for someone to pick up this mutation and have a look and see it, you know, on a computational, you know, from a software, approach, does this look like it would interrupt the function of that protein? So that's not really a test. That's someone in academia, you know, knowing the mutation, and then being, and you know, so therefore being linked into a genetic lab. And you know, sadly, if you're seen by a, if you live in a region that doesn't have those links, then it can be difficult.
Yeah. I mean, what's encouraging is there is more and more of that work going on, and we've got so much better at reading genetic sequences and understanding and doing this work. So, and as more and more people get involved in research, then there's more data, and more people have genetic tests, then there's more data, and this all helps researchers learn. But,
absolutely. And what we do have now is we are making repositories for all these things, and therefore anyone around the world with an interest in, in VUSs, in say in RP, can access all these VUSs. And so it just takes eventually a computational biologist will be able to see the mutation that you have and do it himself. He doesn't need to be, as you say, it doesn't have to have a physical geographical link with the ophthalmologist that is sent the blood off. So yeah, all in time. It's just incredibly frustrating, I do know, for people with RP to have to wait for this all to happen, but that repository is in motion, and that will really help. Huge collaborative effort is required.
Yeah, thank you, and thank you everybody for your questions. We don't have any more on the go. Raj, whose question you answered earlier, has just said thank you very much for explaining it so clearly, and thank you for your talk. So I think that is probably that, and it's bang on 8 o'clock. So Roly, once again, thank you so much for such an interesting talk. It was really good, and thank you everybody for joining us and asking all those great questions, and we really hope that you've enjoyed your hour with Roly.
And thank you very much for having me yet again, and also just for everyone who's tuned in. Yeah, it's very nice to to tell people about what we're doing, and so so thanks for listening.
Okay,
thank you very much, Roly. It's really great, as Kate says, to hear sort of different approaches, and we'd love to welcome you back in the future to hear an update as your research progresses. I just want to thank everybody who's joined us, just to follow on from Kate, but also just to let you know that over the next couple of months, we've got a number of online events planned that you may also be interested in joining. On Wednesday, the 29th of September, we have an information evening. It's a slightly longer 2-hour event, and whilst it is focused for people in Northern Ireland, there's a host of interesting speakers and subjects that will be interesting to anyone, no matter where you are. So please do consider joining us for that. On Wednesday, the 13th of October, we have a similar event that is Wales focused, but again, you're welcome. And finally, on Wednesday, the 27th of October, we'll be hosting our next information webinar. And then we'll be joined by Michael Gilhooly with a fascinating presentation on optogenetics. Over the next couple of weeks, over the next couple of days even, we're going to be sending out an email that has details of where you can listen to Roly's presentation again, just in case you want to just go over it and check any information, share it with people that haven't been able to come along. I'm sure you'll find it as interesting as you do. And we'll also be seeking your feedback on today's session. We really do value your feedback, and it does help us to develop our future webinars and also our other services. So please do reply to that. Thank you all again. Goodbye.